So this is a 33-page review. Most of it I did paraphrase from the book, from the textbook, and added some pictures. But I think it condenses what you went over for the first exam and what's most important. Obviously, the formatting might be different, but the topics are the same. But first of all, any questions you guys might have before we begin? All good? All right. So pretty much physiology and anatomy, they're defined differently, but when you look at them, they are very interrelated. Physiology relates to how the processes of a structure work. For example, if you're talking about the heart, then when you're talking about anatomy, you might be talking about the valves, the mitral valves, the septums, the ventricles, and atria. But the physiology will relate to how the heart pumps blood, how the heart might go wrong, and under pathological conditions. So those are just pretty basic terms when you begin the course. Then we go into the levels of organization, how things are organized from smallest to biggest. You have atoms, molecules, cells, tissue, organs, and organ systems, organisms. Pretty basic. And one acronym I like to use is AMC theaters are open. Then you go into the organ systems, different types. Right here is a representation of the body. It consists of a muscular system, musculoskeletal system, respiratory, digestive, urinary, reproductive, nervous, and endocrine, and immune. I think for each one, you guys are only going to go over integumentary, musculoskeletal, digestive, and reproductive. And then EABS 2 will cover the other half. Broad themes in physiology is homeostasis. You hear this a lot in biology as well. It's the same thing. It implies that all living organisms, they need some sort of balance, either by compensating internal or through external influence. So one example of this would be lizards sitting on rocks on a hot day. Lizards are cold-blooded, whereas humans, which are warm-blooded, but due to their behavioral habits, they're able to achieve this internal homeostasis. Just going forward, going straight into experiments and observations. When you do an experiment, the ideal experiment you want to do, and he does ask this question, is a double-blind crossover. Now, the reason why the double-blind crossover stands above the other ones is because it's a study where neither the scientist, the patient knows that which group they are in, if they're in the control group or the test group, and halfway through the test, they switch them up. They switch the control and the test group. So this is a study design of how a double-blind crossover would look, and you can see that right at the halfway point, they're going to switch the different test groups and control groups to make sure that no bias is introduced. And bias can be introduced in many ways, resulting in type 1 errors or type 2 errors. So the tendency of biases can also be influenced by placebo and nocebo. It's when you take the sugar pills versus the actual pills, or when someone tells you that a certain drug might have a negative effect, which will lead someone else to believe that it does. Other types of studies that he does mention, Dr. Brown in class, includes the meta-analysis, the longitudinal perspective and cross-sectional, and retrospective. The best one out of all of these would be the meta-analysis because it attempts to standardize results from similar tests. Longitudinal is good over long periods of time, but confounding factors might influence the results. Prospective attempts to look back and draw results from previous data. Retrospective. Prospective recruits the participants as they come in, and they attempt to look at outcomes. So chapter 2 relates to molecular interactions, pretty basic chemistry. Just to review on atoms, isotopes, what are they? Atoms have electrons, neutrons, protons, and an isotope has different neutron numbers. The bonding types are ionic and covalent, which lead to different polarities in the case of covalent bonding. Polarity is just happens when you have different electronegative charges, leading to the tendency of electrons to gravitate towards a specific area in the atom or in the molecule. And here's an example of it. Water would be the most ideal example of a polar covalent molecule. And nonpolar would mean that the net distribution of it is equal to 0. So there's no zones where there's slight electronegativity. The reason why this is important is because the body is obviously made up of water, and hydrogen bonding would play an effect to those dynamics. So if you take a drop of water, you'll see that it creates a surface tension, and that's because of the hydrogen bonding. Now, in cases where you might have unpaired electrons in an atom, you get free radicals. Free radicals causes judgmental situations to the body, as it is thought to promote aging later in life. And the one way to mitigate this is by taking antioxidants in your diet. Then we go into radioactive decay and isotopes. The main types of radioactive decay are alpha, beta, and gamma. And this chart summarizes what happens when you have alpha decay. Alpha decay, it emits a helium particle, and beta decay loses a beta particle from the original cell, which is the parent, to the daughter cell, which is the outcoming cell. Gamma emission works a bit differently. And Dr. Brown does mention half-life, which is something you should know, is the amount of time that it takes for half of the original concentration of a molecule to decay. So going to biomolecules or macromolecules, those are important because they can make up the body. So the basic ones are carbohydrates, proteins, and lipids, and nucleic acids. And we can see those in what we eat and how our structures are organized. So the first one we're going to go over is carbohydrates. Carbohydrates consist of monosaccharides, which is the most simplest unit that you can have, and it builds up on that. So monosaccharides convert into disaccharides, convert into trisaccharides, and carbohydrates have a tendency to form either rings or sheets. So the most basic one is glucose, which is the one that most people know. One second. Which is the one that most people know as glucose. But there's also sucrose, maltose, lactose, and fructose. And those are just variations of the rings and the bonding that occurs at the rings. When you go into and stack up these simple sugars into complex sugars, you develop glycogen, which is the one that we have in our human body, and cellulose in plants, and chitin in animals and certain insects. Then we go into lipids. The lipids are different than carbohydrates in the fact that they contain less oxygen and they are nonpolar. So if you compare water versus a lipid, they will have different buoyancies, and that reflects the molecular structure and polarities of those molecules. They're classified into two general categories, saturated and unsaturated. And the most basic unit of lipids are fatty acids. Those fatty acids tend to just go on in chains. At the end, there might be something called a kink, which is just a bend in the chain. And the bend in the chain indicates where there is a saturated fat or an unsaturated fat. Dr. Brown likes to ask about the lipid-related molecules and their structures, and as well as their origin and purpose. So eicosanoids, steroids, and phospholipids, you should know where they come from and what they contribute to. Cholesterol is one of them. So cholesterol comes from steroids, and prostaglandin is an eicosanoid that you'll see in a later chapter why it's important. And phospholipid is important from the cell membrane. So proteins make up the building blocks of our muscles and other enzymes. They're important because we don't manufacture them all. we have 20 that are naturally produced, and our body is missing 9. So these 9 are called essential proteins or essential amino acids because we cannot obtain them without food. So in terms of the molecular structure, they might have amino groups. So nitrogen is going to be included. If you compare the molecular structure of a lipid and a carbohydrate versus a protein, proteins will have nitrogens, which are the designated R amino acid groups. The most basic unit of proteins are amino acids, but when you form them together, they form a peptide bond. And so depending on how you form those peptide bonds, you can link these amino acids together. And these are just technicalities. If you have 2 to 9, you're going to have an oligopeptide bond, and 10 or more is called polypeptide. But this would be the most simplest structure. And when you compare the structures, this would be called the primary structure, which is just one strand of proteins, of amino acids. Then when you combine them, then you get secondary structures, which form either helices or sheets. So these helices and sheets are just the second structure. And when you combine those, you get tertiary structures and quaternary structures when you have multiple tertiary structures. One example of that would be hemoglobin. It's made out of 4 tertiary structures, so 4 subunits. It's very famous for that. Well, not everything in the body is consisted strictly of carbohydrates and proteins. Most of them are a combination of both elements. So we see combinations that form into lipoproteins, which is lipids and a protein, glycoproteins, which is glycogen and a protein, or glycolipids, which is glycogen and lipids. And so those are found at different parts of the body that contribute to the properties of cell membranes and cells. And there's nucleotides. Obviously, there's DNA, RNA, and cyclic AMP, but the most important one in terms of energy is ATP and ADP. They're made out of purines and pyrimidines, which are just the name of the nitrogen bases that they have. And they have a 5-carbon sugar. This is how it looks like. Any questions so far? You guys are free to stop me if you guys have any questions. All good? Hands up? Thumbs up? All right. Now, when we go into nucleotides and you look at the structure and you look at DNA specifically, it's a double helix. So a double helix, yes, it has nucleotides on opposite ends, but it is bonded in between them with hydrogen bonding. And so that hydrogen bonding is a weak bond that allows us to manipulate the structure of DNA, which will be useful for protein synthesis later. Acids and bases, and pH and buffers. He briefly goes over this, but it's more important when you consider the fact that these are conditions that affect enzyme production. So an acid and a base just tells you the concentration of hydrogen ions, and it's measured in a pH scale, which is logarithmic. So I'm going to enzymes. The reason why enzymes are important in the body is because they lower the activation energy. So if you compare a chart dictating the activation energy for a specific reaction, you'll see that when the chart is without an enzyme, it will require a much higher initial energy output versus with an enzyme. And so that's important because it will basically, it's less energy the body has to consume to initiate a reaction. So enzymes, the way they work is usually the protein will have a specific substrate, which is the chemical reaction that it binds to, the chemical it binds to make a product, and it has a substrate site, a substrate binding site. And so the substrate goes to the substrate binding site and forms a product. Now, factors that would influence enzymes would include pH, temperature. It would also include allosteric factor, which is this one. Basically, an allosteric factor means that it needs to have another substrate enter a different site in order for the intended site to show up. So I would call it as an intermediate step. You need A to happen for B to happen. Cofactors is when you need to have 2 things happen at the same time in order to create the substrate. So this cofactor needs to come and bind in order to create the substrate product. Inhibition is when one binds and it prevents the other one from binding. And allosteric factor is when one component binds and the other one is shut off. So chapter 3. All right. Any questions so far, guys? All good? All right. Chapter 3. He does focus a lot on this chapter. So if the first two were just review, but this is really where he takes a lot of the questions from what I remember, because it talks more about the physiology and the anatomy aspect and applications. Whenever he talks about disease, make sure to take note of it, because he usually will mention those diseases in the test, like the Tay-Sachs syndrome we're going to talk about. So the body is compartmentalized into different sections. Here's a region. You have the cranial, pleural, pericardial, diaphragm, abdominal, and pelvic. And so they're separated by membranes. For example, you're going to have the pleural membrane in the lungs and the pericardial membrane in the heart. And so those bodily fluids, when you look at it, it's going to be consisted of intracellular fluid, extracellular, interstitial, and plasma. So intracellular is pretty easy. It's what's within the cell, and extracellular is what is outside the cell. But we can classify these categories into two subcategories: interstitial, which is 75% of the ECF, and plasma, which is 25%. And so when you look at these conditions in osmosis and sodium, or let's say potassium distribution in a cell, the concentration gradients matters, because it will affect the water distribution within the cell. And so the reason for the cell membrane is important. It's pretty easy. You get physical isolation, so you're separating two things from each other. You're regulating the exchange. You're communicating between the inner and outer environment, and you provide structural support to the cell. This is a diagram of how it would look like. You're going to have the phospholipid bilayer, and so that phospholipid bilayer creates vesicles and micelles, which are useful for transporting things in and out of the cell, as well as preventing transport due to the polarity and non-polarity of the head and the fatty acid tails. Also, other things embedded within the phospholipid bilayer is cholesterol and other glycoproteins. And this is just a summary of which ones they are. So other components found within the cell is the cytoplasm and the nucleus. And so the cytoplasm is where you see the specialization of different organelles, which are useful for the production of proteins and for transportation. The mitochondria is useful for energy. Then we have the endoplasmic reticulum, the Golgi apparatus, lysosomes, perioxysomes. And these are just some of them, some of their functions. Most emphasize the mitochondria because it is the powerhouse of the cell. It is double membraned, and it has its own DNA that is different from us, from human DNA. It is ring-shaped. So the reason why it's ring-shaped is that because of the one cell engulfing another cell and leading for that double membrane function. The leaflets are called cristae, and that's where cell respiration occurs. Then we have the Golgi apparatus, which packages proteins, and the endoplasmic reticulum, which packages, this one also packages proteins, and the Golgi apparatus packages lipids. There you go. And the nucleus is where the DNA is stored and is replicated when it comes to mitosis, when it comes time to mitosis. The cytoskeleton, he does pull up a lot of questions on this on the exam that I took, which was 1 year ago. He did some sort of multiple choice or fill in the blank where he gave the different cytoskeleton components and the different proteins. For example, the actin myofilaments, the intermediate lamins, and he asked you what was their purpose and where they are located. So some of them are located in the extracellular matrix. Some of them are located within the cell. So those are important. Motor proteins are able to convert the energy into directed movement. And the mitochondria, we talked about those. So if you want to, you can review these on your own time. Just pretty basic. And protein synthesis, it begins in the nucleus. So I think chapter 4 covers it more in depth, but it begins in the nucleus, and then you have mRNA, which is messenger RNA, and we'll get the strand that from DNA and take it out of the nucleus, put it into a ribosome, which is ribosomal RNA, and create a tRNA. And from that, it's going to go into the endoplasmic reticulum and then be packaged in the Golgi apparatus or in a transport vesicle, which will eventually either go within the cell, just another part within the cell in the cytoplasm, or be excreted outside. Here's another diagram of how it occurs. The major players is mRNA, tRNA, and rRNA. And first you have transcription, translation, and then termination. So for the tissues of the body, you're going to have epithelial, connective, muscle, and neural. And this is a chart. He does test this chart on where it's located and its purpose. He also likes to test on these junction types. The junction types are important for knowing the regions of where they might be applicable, and also for what types of proteins are found at these junction zones. For example, cadherins, integrins, supercams, superfamily cams, and selectins. He does test those. So if you remember this chart, understand where it's located, and are able to talk about it and discuss about each one, then you're in good shape. This one summarizes the different types of epithelia. So exchange epithelia are very flat, are very thin, and they're just used to transport different gases or metabolic waste through the lungs per se, and lining of the blood vessels. So capillaries will be one good example of that. Transporting are columnar or cuboidal, and they have tight junctions because you want to have tight junctions to prevent the movement of the cells. So it'll be very rigid. Then they're ciliated, and ciliated increases surface area, which allows for transportation. A good example of this would be the nose, trachea, and upper airways, also the digestive system. But the digestive system also secretes, mucus, so that would be a combination of both. Protective would be just as it is, it protects the body from foreign invaders or foreign pathogens. The skin is a great example of that, and circulatory. And those are definitions here in more detail. Important for the circulatory, it can be further classified into endocrine and exocrine. Endocrine is just, it stays within the body. So whatever is produced goes within the body, just a different region, and exocrine goes outside from the body. And you have the different connective types, the different connective tissue types. Loose connective, dense irregular. This chart, you should know, he will test it as well. And adipose tissue. And for tissue remodeling, the big thing you should know for tissue remodeling is that there's two main types of endings for cells, and those are apoptosis and necrosis. Apoptosis is basically when the cell has reached its end, its natural end, and programs itself to die. So when it programs itself to die, it starts to shrink, and surrounding lysosomes in the cytoplasm will eventually digest it. And it's just reused for further energy within the cell or within the body. Necrosis, however, is actually pathological and pathophysiological, and is not good because the cell will start to create an inflammatory response. And once it does, it will actually begin to swell. So if you compare quickly the apoptosis cells versus necrosis cells are much bigger, and they explode. When they do, it basically ejects all its contents into surrounding cells, causing further inflammatory response. And so that necrosis is what leads is what is seen in certain types of cancers or certain types of melanomas, which is obviously not good. Any questions so far, guys? All right, chapter 4. It goes over important exergonic and endergonic reactions. This is a chart of how a chemical reaction will look like. It talks about the activation energy, which is the energy you need in order to initiate the reaction. And an exergonic reaction, since you release energy, therefore the products have less energy than the reactants. And in an endergonic reaction, since you absorb energy, the products have more energy than the reactants. And the reason why we use enzymes is because we want to lower this activation energy. So if you compare a chart with and without, this is what I was talking about earlier, you'll see that the one without enzymes is going to be much higher. And so obviously it was more favorable for the body to have less energy because you can use that for other things in the body. Here are some important enzymes that you need and what pathophysiological conditions might arise if you have a deficiency or an abnormality. So he does test this, and he also does test Tay-Sachs syndrome, which occurs when you have a deficiency in, let's see, it's above here somewhere. A deficiency, I think it's in a deficiency in the liposomes, in the lysosomes, or in the lipoproteins. That will lead to the lysosomes to eventually digest itself. Here is the enzymatic reactions. There's oxidation reduction. Then there's hydrolysis dehydration, basically when you add water or remove water. Oxidation reduction is a transfer of electrons, so whether you gain or lose an electron, or whether you gain or lose oxygen in a reaction, and they occur simultaneously. And then you also have basic chemical reactions like exchange, so double displacement, single displacement, addition or subtraction. And ligation is when you join two substrates using energy from ATP. Cell respiration is a big one, so you should know in detail the different steps. There's glycolysis, there's a citric acid cycle, also known as a Krebs cycle, and there's the electron transport chain. And you should know why it's important which one is the most important and its locations in the cell. So the glycolysis occurs in the cell cytoplasm, and then you have the Krebs cycle and the electron transport chain, which occur within the mitochondria. And these are examples of aerobic respiration because they require oxygen. When you look at anaerobic respiration, you're going to have a different mechanism without oxygen. And so the trade-off for having that mechanism is that it's going to produce significantly much less energy. And the currency for that is ATP. So we measure the energy in terms of ATP. That is how the body uses it. In the electron transport chain, it would produce around 30 to 32 total net ATP when you look at the entire thing. This is a chart that compares both processes. When you have anaerobic respiration, you're only looking at glycolysis. So you're stopped at the possibilities of producing the eventual 30 to 32 because you're only producing 2 ATP instead from glycolysis. And so other side effects of glycolysis is that you're going to have buildup of lactic acid. In the case of humans, and in the case of other organisms, you might build up ethanoic. So you're going to build up ethanol because they undergo ethanoic respiration. That's good if you're fermenting beer, but not good when you're hitting the gym. So just some things to consider. Any questions so far, guys? Honestly, anything at all. Questions about future tests, what's going on right now, how you think the exam's going to be formatted, because we reached the end of the line. We're out of things to discuss for this review.